Our final speaker is a graduate of Xavier University Ateneo de Cagayan. He is a registered chemist and is the Assistant Vice President for Administration QMR and Lead Auditor of the Internal Quality Audit Team QC Manager at Ensign Laboratories Incorporated. Here to talk about quality practices in pharmaceutical quality control laboratories, let's welcome Mr. Ray Daniel Reveche.
Good afternoon. This afternoon, I'm going to discuss about the quality practices in the pharmaceutical and the veterinary quality control laboratory. The FDA of the Philippines considered that there should be no delineation between the pharmaceutical laboratory and the veterinary laboratory when it comes to quality control, for the simple reason that the byproducts of the livestock farms becomes part of the food chain of human. So it is just proper that the standards that should be set by any veterinary laboratory and the manufacturers of veterinary drugs and medicines in general must be the same as that of the pharmaceutical. So when we say quality, what does quality mean? It says it is the degree to which a set of inherent characteristics of a material fulfills requirements. This definition is according to the definition given by the ISO 9001:2015 standard. So it means that a material has a set of specifications that is very characteristic to a particular material, then that should be met at all times. When all those qualities or those characteristics are satisfied through testing or any validated method that has been used to assess the material, then it is of quality. Others define it as the sum of all procedures undertaken to ensure that the identity and purity of a particular pharmaceutical or veterinary product is established. So this starts from the time that the material is procured from any accredited external provider. It goes through the standard procedure of sampling and testing, whether it's physical testing or chemical or microbiological. And then the definition given by the World Health Organization is that quality is a part of GMP or general manufacturing practices that is concerned with the sampling, the specifications of the material, the testing that will be employed in that material, and with the organization, meaning the organizational structure, who are the people responsible, what are the qualifications of the personnel involved in quality control, also about the documentation, all records, procedures, protocols, and any implementation to evidence that the quality of a material has been established must be present. And that also includes the release procedures, which ensures that the necessary and the relevant tests are all conducted before any material or product has been released for use. Before it will be used for sale or supply, it must not be dispensed or distributed until the quality has been established. When we speak of quality, there are two dimensions of quality that we consider in particular. So for this case, we have the product quality dimension and the service quality dimension. But since I am more of the product quality, I will talk more of the product dimension. So when we speak of performance, we refer to the efficacy of the drug or that of the medicine. When we speak of features, we are referring to the specific characteristics of a product or that of a drug that would be beneficial to the user. Flexibility, I will be focusing on the formula flexibility. A drug could be in various dosage forms, but there are also drugs and medicines that exist only in one dosage form. If ever a client needs a particular drug that must be in liquid form, it can easily be converted to a liquid form. That means that it can be modified in any way when it is needed. Durability is not much applicable to drugs and medicines. When it comes to conformance, we refer to the compliance to the standards at specifications set by the company or that of the drug manufacturer, as well as to the guidelines set by the regulatory bodies. In this case, here in the Philippines, we have several regulatory bodies. We have the FDA, We have the BAI, or the Bureau of Animal Industry, and we also have the Fertilizer and Pesticide Authority. When we speak of serviceability, we refer to the ease of drug administration, the ease of storage, the ease of transport, the safety and stability of the product. Next is aesthetics. Aesthetics is very important. Okay, we create impressions with the aesthetics. Aesthetic is a response that is immediate and involuntary. Okay, the manner of product presentation is aesthetics. It is a part of product development, and it is concerned with how our senses respond to a product when we see them or when we attempt to buy them. Last one is perceived quality. Perceived quality refers to customers' opinion about the overall quality or image of the product or the brand itself with respect to its purpose or use against its alternatives. Okay, what is quality in pharmaceutical laboratory testing? The right test result at the right time, on the right sample from the right material or product, with result interpretation based on correct reference data. So when we say right test result, we are referring to the accuracy and the precision of the result, that the result is reliable and believable. When we say right sample from the right time, from the right material, it means it is a true representative of the bulk or batch of that product. Right material, we refer that to that material as it has been verified in terms of its identity, and the material is uncompromised against contamination and or cross-contamination that will give positive false results. When we speak of correct reference data, it means that the reference is taken from a material or a reference that is internationally accepted. Like, for example, the USP or the United States Pharmacopoeia or the British Pharmacopoeia. If a company or a laboratory is using a company monograph, then that company monograph must be validated. It has to go through process validation, or in particular in laboratory, that analytical testing procedure must be validated in order to consider that the result is reliable. There are three E's in qualities. The first E that we consider is effectiveness, meaning doing the right things. That means that the extent to which planned activities are realized and planned results are achieved. So the planned activities must be based on validated procedures or methods. The planned results must be the expected output or the outcome. The second E is the efficiency, meaning doing things right. There has to be a relationship between the result achieved and the resources used. Efficiency is using the right technique or method, using the right analytical instrument. The last E is excellence, doing the right things right, meaning you carry out the procedures and methods as accurate as possible. Very, very close to what is exact. Quality control. There has to be an independence of quality control from production is considered fundamental. Independence means there has to be an impartiality. The findings and results must be free from all possible influence or bias. The results are derived according to the objective set by the quality control department. Next one, the quality control manager or the quality control head should have an appropriate qualification and experience which has control over one or several laboratories. Currently, the Food and Drug Administration, even the Bureau of Animal Industry, and even certifying bodies like ISO for ISO certification or for the accreditation of their laboratory as an ISO 17025, the minimum requirement for the quality control manager or the quality control head must be a licensed chemist. The remaining quality control analyst must be at least registered laboratory technician. Next one is there has to be adequate resources. It must be available to ensure that all quality control arrangements are effectively and reliably carried out. When we speak of adequate resources, we refer to the availability of all necessary and basic laboratory instruments and equipment, and it must meet and satisfy the GLP requirements or the good laboratory practices and or the ISO 17025 requirements. Quality control in pharmaceutical laboratory. So this is more specific now with the pharmaceutical laboratory. For ISO, it means that to consider that a pharmaceutical laboratory has indeed a quality control, all activities and techniques applied to ensure that products and services consistently fulfill the requirements. For the product research, there has to be a systematic verification and validation on the compliance of the trial process and reliability and credibility of data. And that, lastly, all measures taken, including the setting of specifications, whether it is the specification of a raw material, a packaging material, or that of the finished product, the sampling method that is being employed, whether it is random sampling or non-random sampling or the military standard sampling, the testing and the analytical clearance to ensure that raw materials, again, the packaging materials and the intermediates or the intermediate process products and the finished pharmaceutical product or it may it be a veterinary product conforms with established specifications in terms of identity, the strength, purity, and other characteristics and complies with the regulatory standards. What are the components of quality control? First, there has to be an adequate facility. I have mentioned a while ago in the from the previous slides that when we speak of adequate facilities, it has to meet and must comply the GLP requirements accreditation, and then including the accreditation requirements of ISO 17025 if you intend to do so, and also meeting the requirements of PRC, FDA, BAI, and FDA. There are certain laboratories here in the Philippines that they are being accredited by the Bureau of Animal Industry and FDA. And there are also other laboratories that for extra credit to them, they subject their laboratory to PRC accreditation or the Professional Regulation Commission. Okay. Next, the personnel must be trained, not just having the license, not just having trainings, not just having continuing education, but what's more important is that all analysts in the laboratory must pass through the proficiency testing. What are the advantages and benefits of passing the proficiency testing? First, if all analysts pass the proficiency testing, then you are able to comply with the laboratory accreditation requirements, whether it is FDA, PRC, or even ISO 17025. The proficiency testing is also a way of evaluating your analyst's performance. And then, from the results of the proficiency testing, you will be able to determine what continuing education and training of staff is being required or must be employed. Next, it ensures customer the confidence in the laboratory results being given to them. And then through proficiency testing, we will be able to identify the problems and areas of improvement. And lastly, you are able to demonstrate the competency to third parties and regulatory bodies. So the next item for the components of quality control is there has to be an approved procedures available for sampling, for inspection, and testing for starting materials, the packaging materials, the intermediate bulk, the finished product. And then lastly, there has to be a monitoring of the environmental conditions for the GMP and GLP purposes. It is very vital that the environmental condition of the laboratory where tests are being conducted must be monitored. All instruments where testings and analysis are being conducted must be subjected to a controlled temperature. The relative humidity must also be maintained or controlled. Because if these analytical instruments are not being operated under their recommended environmental conditions, then there could be possible problems or fluctuations in the results. For the components of QC, we have several components to be considered. For quality control, you have the QC labs, you have the sampling, you have the retained sample, you have the batch inspection and sampling, you have validation, you have maintained and retained documents, and the analysis of finished products. For QC labs, there are several factors that we have to consider. The first one is the chemical testing laboratory. In this laboratory, chemical testing is being conducted in order to tell us what a certain material or product is made of. Chemical testing laboratory can determine how well a product performs, the job that is being designed to do, and how long it will last with normal use. One of the main aims of the chemical testing laboratory is to check the quality of materials by identifying what they are made of and whether they contain anything that shouldn't be there according to relevant standards, requirements, or regulations. The second one is the instrumental analysis laboratory. In this particular laboratory, it encompasses a wide range of instrumentations whose principal purpose is to qualitatively and quantitatively analyze the samples, the chemical makeup of a sample, and the quantity of each component within a sample. The third one is the microbiology and the toxicology laboratory. In this laboratory, we do microbiological testing and toxicological testing. So there are certain antibiotics that are being conducted in the microbiology lab, such as gentamicin for one. A toxicology laboratory is a specialized laboratory environment in which drug tests, also known as tox screens, are being conducted to evaluate the presence of illicit substances. Therefore, the design of this laboratory requires exceptionally air-intensive environment. So there has to be an placement of fume hoods and biosafety cabinets. The fourth one is retention rooms. There has to be a room dedicated for the retained samples. It is practice that there has to be a retained sample for every batch that is being manufactured. The fifth one is the stability room or the stability chamber. In this room or chamber, certain samples of products are being placed, which is being subjected to study. So for products that needs to be further developed, it has to be undergoing a stability program. FDA, for the renewal of the certificate of product registration, you are being required to submit stability test results. So we are being required to submit like the accelerated stability study, meaning the product is being subjected to a high temperature that is 40 degrees Celsius. If it is a long-term stability study, then it has to be controlled in 30 degrees Celsius. So the next one is SOPs or the standard operating procedures, the work instructions, and relevant references. Being in a quality control laboratory, all test methods must be according to a standard operating procedure or a work instruction for purposes of consistency and standardization of the system. And then the presence also of the relevant references such as USP or the BP. Okay. Then the seventh is the documentation room. There has to be a proper place wherein you do the documentation when you do the computation of your results, whether it is qualitative or quantitative. And then the last part is your trained laboratory staff. The laboratory staff must be completely trained aside from having the right educational background. When we talk of sampling, there has to be specific standard operating procedures for sampling before the sampling, during the sampling, and after the sampling. Then there has to be a separate SOP for the sampling of raw material, for the sampling of the packaging material, and that of the finished product. And then there has to be an approved method for sampling. whether it is random or non-random or the military standard method. There has to be well-trained staff and sampling methods to conduct the sampling. And the next one is that there has to be maintenance of all sampling records. It is very important. All sampling records must be maintained and kept for future references if in case there will be questions in the decision of whether a material or product will be rejected or must be passed. And then the next one is that there has to be a checklist for every stage. The purpose of having the checklist is to ensure that no step or process has been left out. Okay, for the validation. Validation is a process of confirmation. You confirm through the provision of objective evidence that the requirements for a specific intended use or application have been fulfilled. This is according to the definition of the ISO 9001:2015. So, as I've said, a process or a procedure must be validated before it can be used in the laboratory. So, if a process or a procedure is a company monograph, as I've said before, then it has to be validated. But if your procedure or your method is taken from a reference like USP or BP, then there's no need for that process to be validated. In validation, all the methods used by QC, such as testing, sampling, inspection, and other activities, are already validated. No activity in the laboratory that has not passed through validation process must be used. Validation of testing method is important and necessary for reasons of consistency, accuracy, and precision. Usually, a validation team or the validating team is composed of technical group, staff coming from the research and development section, staff coming from the production or the manufacturing department, then of course, the staff from the quality control and quality assurance. When we speak of process validation, we're trying to establish an objective evidence that a process consistently produces a result or product that meet its predetermined specifications. So whether the batch size varies from 10 kilos to 100 kilos to 200 kilos, it would still give the very same quality or characteristic of a product. Next, it will provide a confirmation that a process is leading to the intended process results by objective evidence. There are several stages of process validation. The first one is process design. The second one is research and development, including process validation or process qualification. And then the third one is quality risk management, including continued process verification. When we speak of process design, we're talking details of process to be validated. The quality risk management or the QRM is a systematic process for the assessment, the control, the communication, and review of risks to the quality of the drug product across the product life cycle. When we speak of continued process verification or the CPV, we are assessing that during the routine production of a drug or medicine, the process remains in a state of control. When we say state of control, it means that it is in a condition in which the set of controls consistently provide assurance of continued process performance and product quality. Process capability. It says to drive understanding of what is critical to customer and critical to quality parameters. Compares the actual performance or the voice process against how the process should perform as defined by the specification or the voice of the customer. So when we say critical to customer, we are referring and understanding what your customer needs from your product or service, and then restructuring your process against these requirements and will bring you customer loyalty. When we speak of critical to quality, we describe the key characteristics needed to meet customer demands. Records. Records must be made, whether it is manual or by a recording instrument, to demonstrate that all the required sampling, the inspection, and testing procedures have been actually carried out and that any deviation have been recorded and investigated. With records, it is the primary basis of any pharmaceutical activity, and hence all regulations insist on recording of each and every activity carried out. This helps in tracing the history of a batch produced, and this may be required in future if any investigation is to be made. So currently, analytical instruments in the laboratory do have a record of the test results. It cannot be tampered, it cannot be edited. So during inspection, either by FDA or any regulatory body for that matter, they can check the results compared to what you have reported. So that is protecting the reliability of the results and the credibility of the results. And then it is basic concept of GMP, of the good manufacturing practice. It says here, do as you have written, example, your SOPs and the work instructions, and write what you have done. So you have your retained documents or your records. Let's say because for ISO, if you don't have any evidence, if you have no record, even if you have the procedure, then it is as good as you have not done anything. So no record, no conducted activity. Finished product. Okay. The finished product must contain the components complying and conforming with the qualitative and quantitative compositions of the product described in the marketing authorization. Okay. The components must be of required purity and their appropriate containers and correctly labeled. So if your claim, according to your label, that it is 20%, then that drug or medicine must have at least, let's say, 200 milligram per ml. And if it is in percentage, let's say 98 to 102%, then the potency result must only fall within that range. So that means that you are in conformity to the quantitative composition of the product. Qualitative, if your product says, in its specification says that it is white crystalline powder, then the product must be white crystalline powder. Retained samples. So as I've said, there has to be sufficient samples of starting materials and products that must be retained in order to permit further examination of the product if necessary, especially if there is a product complaint or a product recall. The retained samples must be kept in the final packaging unless the packaging is exceptionally large, like if it is in drums or in craft bags when you had it for distribution for sale. Okay, apart from this, there are other activities of quality control that must be observed at all times. So you continually establish and validate and implement all quality control procedures. For work instructions, you maintain sufficient standards and reagents. So if your procedure is obsolete, then you have to update them. If you have new techniques, new references, new editions of references, and it is now you have to update again your procedures. Second, you have to evaluate, you have to maintain your store references and working standards for substances. The third one, you have to ensure the correct labeling of containers, materials, and finished products. It is very important that you retain the identity of every material in the laboratory, especially there for traceability purposes. The fourth one, you have to assess production conditions and in-process quality assurance checks. So you see, the function of quality control is not solely confined to the four corners of the laboratory. The production processes or in the manufacturing department, it has to be assessed also by the quality control staff. The fifth one, there has to be a participation in the investigation of complaints related to quality of the product. Normally, if there are returned products born out of complaint or due to a reported adverse effects, the quality control laboratory will again assess the product. It will go through again the process of chemical testing, analytical testing, microbiological and toxicological testing. Then the sixth part, participate in environmental monitoring. Let's say, for example, in microbiology laboratory or even in the production of sterile products, it is very important that we do monitor the particle count inside the sterile area. It is very important that in the production of products that has hygroscopic materials like sugar, there has to be monitoring using the thermohygrometer or to monitor the relative humidity and of course the temperature. The seventh one, you have to assess the finished product after evaluation. Consider its stability and its continued compliance to the established specifications. Eighth, you have to assess manufacturing and packaging documentation. So if there are lapses in the manufacturing and packaging documentation, there were steps that were neglected, were not verified, or if there were processes that were conducted but were not recorded, then it has to be corrected and the staff in the manufacturing must be called to implement what is required for the maintaining of the quality control. And lastly, there has to be a conformity to specification of the finished product and the examination of the final packaging. That includes the label to ensure that the product contains or has the right label and that the label has the correct information. So I hope with the concepts that I have shared, I was able to impart something that you could apply to your industry or to your own laboratory. Thank you very much.